Okay, let's talk more about microtubules. What color are they in this image? They're the green ones. They're about 24 nanometers in diameter, which makes them the largest cytoskeletal element. They're cylindrical structures formed by polymerized dimers of alpha and beta tubulin. Each dimer has two GTP bound to it. Also remember that they have a positive and a negative end. They grow together in chains, maintaining their polarity. While their growth is relatively slow, they can collapse quickly when needed. So what are microtubules used for? Well, there's really a lot of things. Basically, anytime you want to move something large around a cell, you need to use microtubules. For example, during cell division, what do you want to move around? This would be chromosomes, since you have to separate homologous chromosomes so each cell ends up with a copy. So microtubules, which are these green lines, are anchored to centromeres and pull them apart. What else can we move around? How about carrying vesicles filled with proteins along an axon of a neuron? Now, as I mentioned before, there's a positive and a negative end, and you need a different motor protein depending which direction you're going. If you want to carry something towards the positive end, which is called anterograde transport, you can attach your cargo to a motor protein called kinesin. And if you're going towards the negative end, which is retrograde transport, you can use the motor protein dynein. In neurons, the positive end of the microtubule is at the distal end of the axon, whereas the minus end is near the cell body. So which motor protein is used to move something towards the synapse? That would be kinesin. And by the way, if you're moving a vesicle filled with proteins along the axon using kinesin, where were these proteins synthesized? If they're in a vesicle, then they were made in the rough endoplasmic reticulum, not in a free ribosome. And remember, in neurons, the rough ER is called the Nissl body. The last function of microtubules I want to cover is their role in cilia and flagella, but I'll save that for the next slide. First, let's go over some things that interfere with microtubules. Some drugs that are used to treat cancer, gout, and parasitic infections are able to arrest cellular transport by inhibiting microtubules. Remember, you need microtubules for cell division, so inhibiting microtubules can treat anything that divides a lot, like cancer or microorganisms. Some examples are mebendazole, which treats helminths, griseofulvin, which is antifungal, colchicine, which is used to treat gout, and vincristine and vinblastine are used for anti-cancer. Now, why does colchicine treat gout? Well, first of all, what is gout? Basically, if you have high levels of uric acid in your blood, it'll crystallize, and these uric acid crystals will deposit in joints, especially the big toe. This ultimately causes recurrent acute inflammatory attacks. For longer-term therapy, you treat patients with allopurinol to reduce uric acid, which I talked about in the last section. But during an acute attack, you can use colchicine, which doesn't affect uric acid, but is basically working as an anti-inflammatory, and you can also use NSAIDs and ice to reduce symptoms. You'll learn more about chemo in the hem-onc chapter, but what do you think might be an important adverse effect of inhibiting microtubules with colchicine, vincristine, or vinblastine? You'll have bone marrow suppression, since again we're preventing cell division. And like it says in your book, you can remember this list of drugs that target microtubules using the mnemonic, microtubules get constructed very poorly. So what happens if microtubules aren't working properly? Well, it depends on the specific problem, but one example is called Chidiac-Higashi syndrome. This is usually caused by mutation in the lysosomal trafficking regulator protein, or LYST, which is normally required for organizing proteins into mature endosomes for microtubule-dependent transport. Since granulocytes need this to make their granules, this defect makes patients susceptible to recurrent pyogenic infections, since macrophages and neutrophils can't make the weapons they need to destroy bacteria. These patients also have partial albinism, since melanin can't be transported out of melanocytes, and peripheral neuropathy, since vital proteins can't be transported to the ends of long peripheral neurons. So as I mentioned a minute ago, another role of microtubules is in cilia and flagella. Both cilia and flagella are made from microtubules arranged in a 9 dimers plus 2 singlets formation. So here I'm showing you an electron micrograph of a cross-section through cilia, and you can see the 9 dimers around the periphery and the 2 singlets in the middle. Now in the last slide I mentioned that dynein is used for retrograde transport along microtubules. Dynein can also be used to bind microtubules for a different purpose, which is to link these doublets together. Then ATP can be used to cause them to slide along each other, which makes the cilium bend. Flagella and cilia have a similar structure if you take a look at the cross-section, but they also have some functional differences. First of all, flagella move in this kind of propeller pattern, since they're trying to generate force that pushes the cell away from where it's pointed. Cilia beat back and forth, since they're trying to keep the fluid outside the cell moving in a consistent direction. Flagella are a lot more common in single-celled organisms like bacteria, but can you think of any human cells that have flagella? Sperm are the first ones that come to mind for me. How about cells that have cilia? Two important ones are cells in the lung that keep mucus moving out of the lungs, and cells in the fallopian tubes, which move the egg towards the uterus. Okay, so what happens if you have an autosomal recessive defect in dynein? This is called Cartogner syndrome, or primary ciliary dyskinesia, and if you have it, your cilia and flagella won't be able to move at all, so all these functions I just mentioned won't work. Both males and females will be infertile, since sperm can't move and the egg won't be pushed along the fallopian tube to the uterus. Patients with Cartogeners also commonly have bronchiectasis and recurrent sinusitis, since cilia are required to sweep particles out of the lungs. And lastly, it's associated with situs inversus, which is a condition in which major organs are found on the opposite side of the body. For example, the liver should be found on the right side, but here it's found on the left. Before we move on, let's see if you remember which direction these proteins go. Dynein is used for blank transport, and kinesin is used for blank transport. The options here, by the way, are either anterograde or retrograde. The answer is retrograde, or towards the minus end, for dynein, and anterograde, or towards the positive end, for kinesin. The plasma membrane separates the inside of the cell from the outside, and also is responsible for mediating interactions between cells. As you can see in this image, it's asymmetric, so one side may not look like the other. It's mostly composed of an even mixture of cholesterol and phospholipids, but also contains some sphingolipids, glycolipids, and proteins. Some proteins, like this one or this one, are transmembrane proteins, which means they have an intracellular, transmembrane, and extracellular part. These include transport proteins and ion channels, and receptor tyrosine kinases. There are also peripheral membrane proteins, such as phospholipase C, which are anchored to the membrane either by binding to another protein or a covalent bond to a membrane lipid. It's important to know that hydrophobic domains of proteins will always be found in the transmembrane region, since they're repelled by the water that is outside or inside the cell. Hydrophilic domains will be found bordering the cytosol, extracellular domain, or within the lumen of a transport protein. The sodium-potassium ATPase pump is a transmembrane protein that helps maintain physiologic concentrations of sodium and potassium in the cell. To do this, it utilizes active transport, which means it hydrolyzes ATP. Since ATP is produced inside the cell, it makes sense that the ATPase domain is on the cytosolic side of the protein. Remember, the inside of the cell has a lot of potassium and only a little bit of sodium, whereas the outside of the cell has a lot of sodium and only a little bit of potassium. Therefore, the role of the sodium-potassium pump is to bring potassium in and sodium out. For each ATP used, 3 sodiums are transported out of the cell and 2 potassiums are transported in. So let's say you have a cell and somehow shut down its sodium-potassium pump. Well, what's going to happen? First of all, sodium will start to accumulate inside the cell, since you're not pumping it out. And whenever sodium concentration increases anywhere, water is going to follow it. So the cell will swell and eventually lyse from osmotic stress. So this pump is really important in all cells, but there are some cell types that make their sodium-potassium pumps work really hard. Can you think of an example? One would be neurons, since every time an axon potential goes down a cell, they need to repolarize the cell. And a second one would be muscle, since even though they're dependent on calcium concentration for contraction, the sodium-calcium translocator uses the sodium gradient to remove calcium from the intracellular space, since sodium wants to flow into the cell, and you can use that energy to push calcium out. The sodium-calcium exchanger, by the way, does not use ATP. It just operates through passive transport. There are a few drugs that inhibit this pump. Wabane does this by binding to the potassium binding site and is used as a poison by hunters in Somalia. Cardiac glycosides are drugs used for arrhythmias and congestive heart failure, and they include digoxin and digitoxin. These directly inhibit the ATPase part of the pump, which then causes sodium to build up inside the cell, which in turn causes the sodium-calcium exchanger to allow sodium to leave the cell. This ultimately increases the amount of calcium inside the cell. More calcium in cardiomyocytes means more cardiac contractility. Alright, time for another flash quiz. Let's see if you can remember those immunohistochemical stains for intermediate filaments. So what cell type is stained by desmin? And the answer to that one would be muscle.